Good afternoon all, and we are just left with two concepts from cell the unit of life, includes cytoskeleton and microbodies. And then once we will revise the entire chapter, go with the objective session. So cytoskeleton, what is the name refers to? The name itself, cytocell, skeleton, the body of the cell. Okay, those structures which help in making up the cell body, which gives mechanical support to the cell and which helps both in the intracellular and intercellular transport in the cell. Okay, those are what? Proteinaceous structures. So what is cytoskeleton? Cytoskeleton includes some group of proteinaceous filaments like microtubules, microfilaments and intermediary filaments. This together will give in what support ma? They will provide mechanical strength, mechanical support or mechanical strength, and they help both in intracellular and intercellular transport, and they also play crucial role in cell motility. First we will look on the structure of microtubules. Okay beta, structure of microtubules. Microtubules are hollow unbranched cylindrical structures. Okay, remember what are they? Hollow unbranched microtubules consist of what protein? Tubulin. So these are hollow cylindrical structures, right? They will be of 20 in diameter. Likewise, a single microtubule is made up of each protofilament will have tubule. Mechanical there second Is the voice clear now? Now, microfilaments, if we observe, functions include, first is cytoplasmic streaming, formation of myofibrils, that is muscle fibrils. We know actin and myosin will play crucial role that we learn in muscle contraction and relaxation mechanism. Next, formation of movement of microvilli in small intestine, and formation and retraction of pseudopodia, especially in amoeboid protozoans like amoeba and entamoeba, endocytosis and exocytosis, and this also helps in formation of spindle apparatus. But then if these three proteinaceous filaments are given and the question is like, what proteinaceous filament is the major constituent of spindle fibers, your answer should be microtubules. If at all this is not given, then your second option should be microfilaments. Okay, be clear in choosing. Next, movement of cell components. Okay, so this microfilaments will highly helpful in motility. Okay, be it with myofibrils, cytoplasmic streaming, microvilli, pseudopodia, endocytosis and exocytosis, and movement of cellular components. Mostly motility is with respect to what? Microfilaments. Next is intermediary filaments. These are also unbranched structures. They are formed by some variety of proteins, but then these proteins are solid. They are solid proteins. And here you see these functions include formation of nuclear matrix, support of membranes, they will aid support in membranes. I already told you, intermediary filaments functions are related with what? Nucleus. They are specifically useful for nuclear transport. And supporting array of cytoplasm, they support of contractile units and mechanical strength to nervous system. They also help in mechanical, they also give the mechanical support to the nervous system. Till here it is clear, right? Okay. Next is microbodies. Microbodies are single membrane bound organelles. They are present in both plant and animal cells. They are small membrane bound vesicles that are seen in cytoplasm of both plant and animal cell. They absorb what? Molecular oxygen, and they take place in oxidation. They play major role in oxidation, especially for oxidizing long fatty acid chains. Okay. Microbodies examples include peroxisomes and glyoxysomes. Glyoxysomes are one specialized type of peroxisomes which are present in germinating seeds. Germinating seeds. And they play crucial role in converting the stored fats to starch via what? Glyoxylate cycle. Okay. What are glyoxysomes? One specialized type of peroxisomes. They are present in germinating seeds. They are helpful in converting fats to starch through glyoxylate cycle that operates in germinating seeds. Clear till here? Here you see peroxisomes, single membrane bound organelles. Glyoxysomes are microbodies which are specialized peroxisomes. Their occurrence both plant and animal cells, but glyoxysomes are present only in germinating seeds and some of the fungi. Functions of peroxisomes include fatty acid oxidation, lipid synthesis, detoxification and photorespiration. You all know that C2 cycle operates only in C3 plants. And functions of glyoxysomes include beta oxidation of fatty acids, that function is common with peroxisomes and glyoxysomes. And next is what? Glyoxylate cycle, that takes place only in germinating seeds. Next, peroxisomes, whose role, what is the role of peroxisomes? General metabolic and detoxification of different cell types, and here for seed germination. Okay, active mobilization during seed germination is conducted via glyoxysomes. Clear na? Peroxisomes and glyoxysomes. In simple terms, you have to remember the functions and occurrence, enough. Both are single membrane bound, the functions are very much important. Glyoxysomes are present in germinating seeds, both of them are helpful in oxidation, beta oxidation of long chain fatty acids, and detoxification, lipid synthesis by peroxisomes, right. So, with this, cytoskeleton and microbodies is also done. So, before getting to PYQs, let us once recall the complete chapter in like 10 to 15 minutes, have some water and sit straight. Yeah, done, right? Fine. So, in the beginning, first come first, about cell discoveries. Whoever remembers, na, be quick in response. First, from cell discoveries, 3 discoveries we have learned. One is Robert Hooke discovered dead cell from what plant? Oak tree. Quercus tuber, what type of cells? Cork cells, and he is the first scientist to observe about cell wall. He did not discover it, but he observed about cell wall. Second is living cells, identified by Anton van Leeuwenhoek. He named the living cells as animalcules, and he is the father of microscopy. Third, Robert Brown discovered nucleus in what cells? Root cells of orchid, right. So, with this, discoveries is done. Next, cell theory proposed by Schleiden and Schwann. Cell theory proposed by Schleiden and Schwann. Schleiden is a German botanist, be louder. Schwann is a British zoologist. And Schleiden has given what point in which year? 1838. All plant bodies are composed of cells and products of cells. Schwann in the year 1839 has said all plant bodies and animal bodies composed of cells and products of cells. Limiting boundary of animal cell is cell membrane. Above that, cell wall is present in plant cell is said by or discovered by whom? Schwann. Though he is a British zoologist, he has said cell wall is the limiting boundary of a plant cell. Then who gave modified cell theory? Rudolf Virchow. What is the point that these two scientists have failed to put it in cell theory? How new cells are formed. And Rudolf Virchow has put it under what statement? Omnis cellula e cellulae. Omnis cellula e cellulae. In the year 1855. Very good. Okay. So, with this, cell theory, modified cell theory is done. Next, exceptions of cell theory includes enucleated cells like mature RBC, tracheids, sieve tubes, and multinucleated cells like rhizopus and viruses, which are infectious agents that does not have cell membrane, cytoplasm and all. It just has genetic material and protein code. Above that, it has envelope. With this, cell theory and exceptions are done. Now, you just recall different sizes and shapes of the cell. Which is the smallest living cell? Mycoplasma. Can you tell me the example, diagram example which is given in our NCERT? PPLO, one example of mycoplasma whose size is 0.1 micrometer in diameter. And tell me the cell shape, which cell shape is amoeboid? White blood cells, round and biconcave. what is the shape of columnar epithelial cells? Long and look at the NCERT and tell me the sizes and shapes of the cells. Recall it na. See here. Long and narrow. Tell me about the longest cell. Nerve cell. Largest cell. Egg of ostrich. This completes sizes and shapes of the cell. And then we have looked into the prokaryotic cell. Cell envelope of prokaryotic cell, outermost layer, glycocalyx. If it is tightly arranged, capsule. Loosely arranged, slime layer. Capsule helps in escaping from host immune system. Slime layer prevents the loss of water and minerals. Second layer of cell envelope is cell wall, which consists of peptidoglycan. Based on the concentration of peptidoglycan, it is of two types, gram positive and gram negative. Gram positive bacteria is composed of 80% of peptidoglycan, good. Murin, teichoic acids. Can you tell me a few examples of gram positive bacteria we have discovered? We have discussed, sorry. Bacillus, Clostridium, Corynebacterium. Gram negative bacteria, how much percent of peptidoglycan? 20%. Along with that, it has unsaturated lipid chains, unsaturated fatty acid chains, and it has polysaccharides. Okay, and it has pseudomurin, teichoic acids are absent. Okay, in Christian gram staining technique, okay, how many dyes are used? 2 dyes, crystal violet and safranin. Which type of bacteria is retaining the crystal violet stain? Gram positive. Which type of bacteria is dissolving the stain and again restained with safranin? Gram negative. Okay, just remember that we may get some statement based on gram positive and gram negative. Most of the pathogenic bacteria are gram positive or gram negative? Gram negative. Most of the pathogenic bacteria are gram negative, remember. So with this, cell wall is done. Next is cell membrane. Cell membrane has some invaginations. What are they? Towards the cytoplasm. Mesosomes. Those extensions can be in the form of tubules, vesicles or lamellae. Okay. So these mesosomes of prokaryotic cell have diverse functions, of which one function is highlighted. What is that? Respiration. That is the reason mesosomes of a prokaryotic cell are equivalent to mitochondria of eukaryotic cell. Could remember? Can you tell me the functions of mesosomes? You can refer your running notes, NCERT, but keep answering and recalling. Cell wall formation, DNA replication, formation of daughter cells, secretion, increase in surface area and respiration. These are the functions of what? Mesosomes. Now entering into the cytoplasm of prokaryotic cell, it has two types of genetic material. What are they? One is genomic DNA, second is non-genomic DNA or plasmid DNA. Genomic DNA is also known as nucleoid DNA or chromosomal DNA, whose main function is what? Conducting metabolic activities. Whereas non-genomic DNA or plasmid DNA, three major functions given in our NCERT, what are they? Resistance to antibiotics, very good. Conjugation, specific phenotypic character to the bacteria. It gives specific phenotypic character to the bacteria. These three are the functions of what? Plasmid DNA. Then we have learned non-membrane bound organelles, ribosomes and nucleation bodies. Ribosomes are what type? 70 is right. Ribosomes consists of what? RNA and proteins. How many different types of proteins are present in ribosomes? 80 different types of proteins. Register in your mind how many different types of proteins? 80 different types of proteins. And this ribosomes adjoining to the mRNA, it is in what condition in prokaryotic cell? Polysome. Polysome or polyribosome. And where are the ribosomes present in a prokaryotic cell? Cytoplasm and attached to plasma membrane. Next, coming to the inclusion bodies. Inclusion bodies are storage bodies which will store the food molecules. Okay. Some examples of inclusion bodies. Sinophycean granules, phosphate granules, sulfate granules, these are the examples. Gas vacuoles are also present, which will help in buoyancy in few of the prokaryotic cells, right. So, with this non-membrane bound organelles also done. Next, looking into motile and non-motile structures of a prokaryotic cell. Only motile structure is what? Flagella. Made up of what protein? Flagellin. Has what microtubular arrangement? 9 plus 0. What are 9 plus 0? What are the three components of flagella? Basal body, hook, filament. Okay, it has basal body, hook and filament. Clear na? And non-motile structures include pillae and fimbriae. Pillae are the tubular extensions, fimbriae are bristle-like structures sprouting out of the cell wall. With this, prokaryotic cell is done. Coming to eukaryotic cell, we will start with the outer boundaries. First with cell wall. Cell wall is of different compositions in different cells. We have also recalled two kingdom system of classification, where all the organisms which have cell wall are placed in plant kingdom. Likewise, bacteria has what composition of cell wall ma? Peptidoglycan. And algae has what cell wall composition? Galactans, mannans, and minerals like calcium carbonate, right. Fungi has what cell wall? Chitin, also known as fungal cellulose. Plants has what composition? Cellulose, hemicellulose, pectins and different types of proteins, okay, right. So, these are different compositions we have learnt. Then, what is the first formed cell wall layer in plant cell? Very good, middle lamella. First formed cell wall layer is what ma? Middle lamella, which is a common wall layer that glues two neighboring cells, right. So, this middle lamella will be formed as initially what? Phragmoplast, that is extended to the periphery, forming out cell plate and middle lamella. That is synthesized from which organelle? Golgi bodies. Remembered? Now, inner to the middle lamella, what is formed? Primary cell wall, which is thin, extensible, made up of parenchyma. Inner to the primary cell wall, what is formed? Secondary cell wall. Secondary cell wall can be 2 to 3 layered. It consists of what? Cutin, suberin, lignin, pectins and all this, right. Next, inner to the secondary cell wall, we have cell membrane. So, now if we see the hierarchy from cell membrane to the middle lamella, we have secondary cell wall, primary cell wall and middle lamella. From out to in, if we observe, middle lamella, primary cell wall, secondary cell wall and cell membrane. That hierarchy or organization in both ascending and descending you have to remember. Next is cell membrane. Cell membrane model, fluid mosaic model, which is widely accepted, proposed in the year 1972 by Singer and Nicholson. According to them, what are the components of cell membrane? From RBC, right? RBC or erythrocyte. What are the components? Proteins, lipids, carbohydrates. Proteins are of three types based on the ease of extraction. What are they? Integral proteins, peripheral proteins, transmembrane proteins, right. So, and then lipids are amphipathic in nature. They have hydrophilic polar head, hydrophobic non-polar tail. Hydrophobic non-polar tail is protected from the aqueous environment by hydrophilic polar head, right. And the quasi-fluid nature of lipids will enable lateral movement of proteins. Carbohydrates are present as glycoproteins and glycolipids. They are being synthesized from Golgi bodies itself, right. So, this is the composition of what? Cell membrane or plasma membrane. Clear with plasma membrane? And we have learned few functions of cell membrane. Do you remember? It helps in cell motility, cell division, formation of intercellular junctions and cellular transport. Cellular transport is of two types. We have three types of cellular transport: passive transport, active transport, bulk transport. Passive transport, three types we have learnt: diffusion, facilitated diffusion, osmosis, from high concentration to low concentration along the concentration gradient that does not require any ATP. Second type is what? Active transport. which requires energy, that is against concentration gradient from low to high, right. So, this requires ATP. Example is sodium potassium pump. Bulk transport of two types, endocytosis and exocytosis. Endocytosis is of two types. What are they? Cell drinking, cell drinking and cell eating. Pinocytosis and phagocytosis. Second type is what? Exocytosis, which is also addressed as cell vomiting. Functions of cell membrane done. Next, we have learnt on single membrane bound organelles, right. First is endoplasmic reticulum. RER synthesizes proteins. SER functions include lipid synthesis, detoxification of drugs, synthesis of steroid hormones, and regulating calcium ion concentration, and SER present in muscle cells known as what? Sarcoplasmic reticulum. Okay. So, ribosomes, what type of ribosomes are attached to RER? 80S, eukaryotic cell, right? It has 80S ribosomes attached to the RER. In 80S ribosomes, which subunit is attached to endoplasmic reticulum? 60S subunit. It is attached via what? Riboforin protein, right. So, next, the protein lipid synthesis functions of ER are clear to you. Now, which phase of Golgi is towards the ER? Cis phase. Inside Golgi bodies, what is happening? Processing, packaging. What are being released out from Golgi? Lysosomes, which are rich in hydrolytic enzymes, glycoproteins and glycolipids. From what side of Golgi? Trans phase, concave or maturing phase. Likewise, lysosomes are rich in hydrolytic enzymes. Can you name few hydrolytic enzymes? Hydrolases, lipases, proteases, carbohydrases, right. And then this will help in digesting the digestive molecules or any dead cellular particles. Primary lysosome attaching to the phagosome, that is digestion particle, is known as what? Secondary lysosomes are heterophagosomes. After digestion, the particles left over are known as what? Residual bodies. Primary lysosomes attached with any dead cellular particle known as what? Autophagosome or autophagolysosome. Recall 4 different types of lysosomes. After this, we have learnt on what? Vacuoles. What is sap vacuole? Vacuole which is present in a plant cell. How much percent? 90% of plant cell volume, right. And half the molecules from cytoplasm enter into the vacuole? Very good. Against concentration gradient via active transport. We have discussed on other types of vacuoles also, like contractile vacuole, food vacuole, gas vacuole. Contractile vacuole will help in osmoregulation and excretion. Best example we can take euglena. Food vacuole we can take paramecium, which will help in engulfing of food material. Gas vacuoles will help in buoyancy. Best example we can take amoeboid protozoans, right. With this endomembrane system done. Next, what type of ribosomes are present in a eukaryotic cell? 80S and 70S. 80S attached to ER and in cytoplasm, 70S in chloroplast and mitochondria. The association of larger subunit and smaller subunit in ribosomes is decided by what ion concentration? Magnesium ion concentration will decide the association of LSU and SSU, that is larger subunit and smaller subunit. Clear with ribosomes? Ribosomes discovered by George Palliday, and endoplasmic reticulum discovered by K.R. Porter. Fine. Golgi bodies? Camillo Golgi. Okay. Few scientist names you have to remember. And then after ribosomes, centrioles. Centrioles have what microtubular organization? 9+0. Central proteinaceous hub, 9 radial spokes. Each radial spoke has 3 microtubules. So overall total how many microtubules are present in centrioles? 27 microtubules. Then we have learnt on cilia and flagella. Has what microtubular arrangement? 9 plus 2. Central 2 microtubules, each radial spoke has 2 peripheral microtubules. Total how many microtubules? 21 microtubules. Okay, 18 plus 2. 2 central microtubules plus 9 radial spokes, each has 2. So 20 microtubules. Okay, so total has how much? 20 microtubules. So with this flagella, eukaryotic flagella has what protein in it? Tubulin protein. Prokaryotic flagella will have what protein? Flagellin. Eukaryotic flagella, tubulin. Remembered? Yeah. So, and then microtubular arrangement you are clear with, and then double membrane bound organelles. Plastids, how many types? 3 types: chloroplast, chromoplast and leucoplast. All are interconvertible. Yes, right? Leucoplast is of 3 types again. If it is storing starch, proteins, alluroplast, lipids, aleoplast. Chloroplast is a double membrane bound organelle. Third membrane system is membranous sac thylakoids. Stalk of that sac is grana. One grana connected to other grana by stroma lamellae. Where are light reactions happening? Inside grana, exactly at thylakoid membrane. Dark reactions or light independent reactions occur in stroma of the chloroplast. Chloroplast is known as semi-autonomous organelle or self-duplicating organelle. The stroma of the chloroplast, can I call it as matrix of chloroplast? What are the components of it? It has 70S ribosomes, double-stranded circular DNA, starch granules, lipid granules, starch granules and lipid droplets, all these are present inside the chloroplast. Next we have learnt on what? Mitochondria, double membrane bound organelle. Amongst outer and inner membrane, which is more permeable? Outer mitochondrial membrane is more permeable, inner mitochondrial membrane is selectively permeable, which has enfoldings, and we will call it as what? Cristae. All electron carriers and enzymes required for aerobic respiration are present in inner mitochondrial membrane. Matrix of mitochondria will contain what components? 70S ribosomes, double stranded circular DNA, RNA molecules and proteins, right. So, recalled about mitochondria, and let us recall the sites of aerobic respiration once. Oxidative decarboxylation and TCA cycle will happen in matrix of mitochondria. ETS will happen in inner mitochondrial membrane, right. And then we have looked into nucleus, has 4 components: nucleolus, nucleoplasm, nuclear poles and nuclear envelope. Nucleolus will synthesize what? rRNA. By what enzyme? RNA polymerase 1. In nucleoplasm, tRNA and mRNA are synthesized. mRNA by RNA polymerase 2, tRNA by RNA polymerase 3. Good. And DNA is present inside the nucleus in the form of chromatin. Condensation of chromatin is via histone proteins by histone octamer. What are the proteins? How many types of proteins are present in histone octamer? 4. What are they? H2A, H2B, H3, H4. 2 sets of them, right? And the wrapping of DNA around the histone octamer will give a nucleosome model. Likewise, a single nucleosome will have how many base pairs of DNA? 200 base pairs of DNA. Clear, right? Recalled everything? Yeah. Now, at higher level of condensation, non-histone proteins also require. So, a single chromosome will have histone proteins, non-histone proteins, DNA and RNA inside it. And then we have looked into structure of chromosome, shapes of chromosome. we have 4 shapes. If centromere is at the center, what shape it is? V shape and metacentric. It is slightly away, giving long and short arms of chromatid. That is what shape? J-shaped chromosomes. Sorry, L-shaped chromosome, submetacentric. If it is very far from one chromatid, one arm of the chromatid, and very close to the other arm of the chromatid, that forms J-shaped and it is acrocentric. If one arm of the chromatid is negligible, that is telocentric. At least 80% of chapter you recalled in peripheral phase. And then just now we have learned, right, cytoskeleton and microbodies. Okay, just take 1 minute break and then we will start the objective. Yeah, look at the first question. This and all we have done till 2025 we have done, right? First this row. The DNA present in chloroplast is circular and double stranded. Single stranded DNA or double stranded DNA? Double stranded circular DNA. Where is it present exactly? Stroma of chloroplast. Is it self-duplicative? Yes, it can duplicate inside the chloroplast. This question to this row. Guess or exactly you are saying? Huh? Okay. Any other answers from this row? No, we will see. Nucleolus is site of what? rRNA synthesis. Centriole, organization like cartwheel. Leucoplast is the body where it stores the nutrients. Golgi bodies is site for synthesis of glycolipid and glycoprotein. Correct, right? Then why so much of confusion? You should be clear with your answer. This row. Good, we will see. Citric acid cycle or TCA cycle, mitochondrial matrix, glycolysis, electron transport system, inner mitochondrial membrane, proton gradient, intermembrane space. What is the right answer? 2. This row. Simple, I have extracted it from biomolecules, but then we have learnt about hydrolytic enzymes here. We need to connect. We are not learning it for first time, right? Lipases will act on what? Lipids. Has what bond? Ester bond. Nuclease will act on what? Nucleic acids, which acts on what? Phosphodiester bond. It has 3 components for each nucleotide. What are they? Sugar, phosphate group and glucose. sugar phosphate group and nitrogenous base. Okay, and they are connected by a what? Phosphodiester bond. Two nucleotides are connected by a phosphodiester bond. Nucleic acid is a polynucleotide chain, right? So protease will cleave peptide bond. Amylase will act on glucose, which has glycosidic bond, right? So what is the right answer? Next. Questions from the year 2024. 4? Got the answer already, we will see. Axoneme is the central part of cilia and flagella. So 2. Cartwheel pattern, you can see it in centrioles. Cristae, mitochondria, inner mitochondrial membrane. Satellite DNA you can see in chromosomes. So what is the answer? Next. Statement based question. How about statement 2? Is correct. Mitochondria and chloroplast are both double membrane bound organelles. True. Statement 2. Inner membrane of mitochondria is relatively less permeable as compared to chloroplast. Correct. That is why you should not be overconfident. Just in a single look. Okay. So see here, in chloroplast we have two membranes, right? Inner membrane doesn't have any complexes like how we have in inner mitochondrial membrane. Inner mitochondrial membrane has 4 complexes, it has electron carriers and all. So it is selectively permeable in nature, whereas chloroplast membrane is mostly semi-permeable in nature. When I am telling you about the synonyms of plasma membrane, I have told you plasma membrane, cell membrane, selectively permeable membrane, semi-permeable. The key difference between selectively permeable and semi-permeable, selectively permeable means it will select in the solute particles or ions, it will select which molecule to enter and which to not. Semi-permeable, which will easily allow the solvent particles and it will not allow the solute particles, right. So statement 2 is also correct. So 1 and 2 both are correct, right. Next, 2023. Next, this question. Whoever want to answer first, just raise your hand. Give space so that others also can think. Two, okay. Others thinking? Second option, okay. Which of the following is not considered as a part of endomembrane system? Mitochondria is not considered. Endoplasmic reticulum, Golgi bodies are under the endomembrane system. Chloroplast is also not under endomembrane system. Peroxisome is, so A, C, E, option 2. Though you are quick at your response, before answering once recheck ma. Okay. 2022. When we are two terms, euchromatin and heterochromatin. At the time of packaging, if it is tight, see. Eukaryotic true. Heterochromatic active. Nucleosome arginine. Shapes of the chromosome easily answer. I would like to hear some synonyms of mycoplasma as well, if you remember. We will give others time. What happened, Toshita? Navadeep, you can answer to me. Mycoplasma is than 1 micron filter paper, true. In other terms, like in your area, there will be a line that these are wall-less bacteria and they can pass bacteria of plant kingdom with cell wall. Wrong, they are without cell wall. So statement 1 is correct and 2 is next. Each year can you see 4 to 5 questions related to this. Of course, purely 3 questions you will get. Connection you will get 2 questions at least. Beings or with different types. Of the following statement, prokaryotes, no endoplasm only. Other 3 are correct. Next, from nucleus. Unfoldings of mitochondria, thylakoids, flattened membranous sacs, centromere, cistern. Right, what is the correct answer? Simple question. Organelles included in the endomembrane system are ER, Golgi complex, lysosome, the time you are getting which simple.